the next question is question number 51. So this question says that which of the following animals has three-chambered heart? Pteropus, Scoliodon, Hippocampus, and Chelone. So this question has been asked from the chapter of your Animal Kingdom of Standard 11. And let me tell you that three-chambered heart is usually found in case of all the reptilian categories of animals. That means all the reptiles. All right. So three-chambered heart is usually found in reptiles and amphibians. So amphibians and reptiles, they usually represents the three-chambered heart. All right. So let us take a look that to which category they actually belong to. All right. Now, so the first category is considered to be as pteropus. So pteropus is a category which is also known as flying fox, and this is a category of this belongs to mammalia class that has four-chambered heart. So this is basically known as flying fox, belonging to class mammalia. Class mammalia and has four-chambered heart. So that means definitely this is not the correct answer. All right. Now, the next category is Scoliodon. Scoliodon is basically a category of fish. So this is also known as dogfish. All right. So this is also considered to be dogfish. And we know that fishes have two-chambered heart. So they have two-chambered heart. All right. And this basically belongs to Pisces. The superclass is Pisces. Superclass is Pisces. All right. Then the third option is considered to be as hippocampus. Now, hippocampus is also known as seahorse, and this again belongs to, this is a category of fish. All right. So, hippocampus. This is called seahorse, belonging to Pisces. It basically belongs to fishes category, Pisces, and has two-chambered. All right. Now, the last option which is left is the Chelone. Now, Chelone is a category that is also known as, or this is an option which is also known as turtle. All right. And we all know that turtle belongs to class Reptilia that has the presence of, three-chambered heart. So this belongs to Reptilia with three-chambered heart. All right, so this is the case. So they have three-chambered heart. So that's why the correct answer is option D over here. All right, now question number 52. Question number 52 says that which of the following types of epithelium is present in the bronchioles and the fallopian tube? Now, this question has been asked from the chapter of your animal tissues of standard 11th. All right. And it is a very direct question that has been asked because you just have to, know this fact that bronchioles are a part of lungs and fallopian tubes are a part of the female reproductive system. All right. And these two structures are unique in the sense because the inner surface of these two structures, they have a very important, the inner lining has a very important epithelium, and that epithelium helps in the, movement of substances or movement of certain structures from one place to the another place. So we can say that out of the all the options that has been given, which is stratified squamous epithelium, simple squamous epithelium, simple columnar epithelium, and ciliated epithelium. So out of the all the options that has been given, the correct answer is considered to be as ciliated epithelium because these structures, they have the presence of cilia on their inner surface. All right. So bronchioles, which are found in lungs, and fallopian tubes, which are a part of female reproductive system. Female, just a second. Reproductive system. So they have the presence of ciliated epithelium. So have the presence of cilia, which are fine finger-like projections or brush border epithelium, you can say. Cilia having brush border appearance. On their inner surface. They usually have cilia, which is having the brush border appearance on their inner surface, thereby indicating ciliated epithelium. Ciliated epithelium. All right, so this is the case. So that's why the correct answer out of all the options is ciliated epithelium, because this cilia, it actually helps in the bronchioles. It will help in the, in filtration of the particles. It will actually trap all the dust particles and will filter out the fresh air. While in case of the fallopian tube, these cilia, they are basically responsible for helping the passage of released ova from the ovary, so that the released ova can easily pass from the ovary towards the fallopian tube and then towards the uterus. All right. So that's why the correct answer is option D over here. Now, question 53. Question 53 says that which of the following is not an intrauterine device? So this question has been asked from the chapter of your reproductive health of standard 12. And again, this is a very direct question, very easy question that has been asked. So let me tell you that intrauterine devices, which are also called as IUDs. All right. So intrauterine devices, which are also known as IUDs. So these are one of the contraceptive methods by which is in which the certain devices that can either be medicated or non-medicated or hormonal, they are inserted into the uterus of the female. So in order to prevent pregnancy. All right. So IUDs are basically contraceptive methods. These are contraceptive devices inserted in the uterus of female to prevent pregnancy. All right. Now the category of these IUDs, they are of various kinds. All right. So IUDs are of various types actually. If we just look into the types, so they can either be non-medicated IUDs, then there is copper releasing IUDs, and the third is the hormone releasing IUDs. Hormone releasing IUDs. So in case of the non-medicated IUDs, the example includes lipase loop. This is an example of non-medicated IUD. So this includes lipase loop. Then in case of copper releasing IUDs, it includes copper T, copper 7 and multi-load 375. It's multi-load 375. And in case of hormone releasing IUDs, it includes Progesta Cert and LNG20. So Progesta Cert and LNG20. So let us take a look into the options that has been given. So Progesta Cert, this is a hormone releasing IUD. So again, this is not the option that is there. So this is hormone releasing. So the question basically is asking the not an intrauterine device. So definitely this is an intrauterine device. So definitely this cannot be the case. Then there is progestogens. Progestogen is basically a contraceptive pill. All right, this is a contraceptive pill. This is not an IUD. All right, not an IUD method, you can say. So that's why this is the correct answer. While multi-load 375, this is an intrauterine device, and lipase loop is also an intrauterine device. So that's why the correct answer is progestogens, which is actually a category of contraceptive pill, which is orally taken by the females to prevent pregnancy, but it is not a category of IUD. All right. So that's why the correct answer is option B over here. Now, question number 54. So question number 54 says it's a match the column question that has been given, and you have to find out the match the listings. You have to choose the correct answer from the options given. So let us take a look with the very first option, which is Chlamydomonas. So we all know that Chlamydomonas is a category of green elega, and this is one such category. As you can see the options that has been given, so these are all the asexual reproductive structures that has been mentioned. All right. So Chlamydomonas usually are responsible. This is a green elega. So a green elega where the asexual reproduction is going to take place with the help of zoospores. All right. So this is going to match up with A, where asexual spores are biflagellated zoospores. That means they are motile. They are motile. All right. So that's why the option A is there. Now the next is penicillium. So penicillium is a category of fungi. This is an Ascomycetes member of fungal category, and this penicillium, it has the presence of the asexual spores which are found in penicillium are the conidia. All right. So in case of penicillium, if you talk about it, the asexual spores are conidia. So asexual spores are conidia. Now the third option is hydra. So in case of hydra, the asexual reproduction takes place through, through the process of budding. Budding is one of the way by which the hydra is going to undergo the process of asexual reproduction. So hydra, it basically produces buds as asexual reproductive structures. All right. Now, the D option is left with sponge. So sponges, they show a very peculiar feature of gammules formation. So sponges, in case of sponges, the gammules are the asexual reproductive structures. All right. So let us match with the correct options that has been given. So A is going to match up with third. Then B is going to, sorry, A is going to match up with second. Sorry. A is going to match up with second. Then B is going to match up with first. C is going to match up with fourth. And D is going to match up with third. That's why the correct answer is option C over here. All right. Now, the next is question number 55. Now, this question says that which of the following reasons is mainly responsible for graft rejection in the transplantation of organs? Now, this question has been asked from the chapter of your Human Health and Disease of Standard 12. All right. And let me tell you that any tissue or organ which is, which is used for transplantation process, that is called as grafting. So the other term of grafting or the other term of transplantation, we can say, is also known as grafting. Now, in case of grafting or transplantation, when it happens, the tissue matching, the blood grouping matchings are very essential. And if in such cases, if there is the rejections are only, the rejections only takes place when there is the cell is not able to accept that particular organ or that particular tissue to the body. All right. And that's why it is a response of uh, basically cell category, cell mediated immunity, but we will also see to the different options that has been given. So the first option is cell mediated response. The second option is inability of recipient to differentiate between self and non-self tissues. Again, this won't be the case, because if this will be the case, then autoimmune disorder will be caused. Humoral immune response only, this is related to blood. Again, this won't be the option. And autoimmune response, again, this won't be the option. So the technically the correct answer is A only, because it is dependent on the acceptance or rejections of the cell. So we can say that any tissue or organ getting transplanted is called grafting. All right. So in case of grafting or transplant, the tissue matching the tissue and blood grouping matching is important for undergoing graft. It is because of the cell mediated immune response that might show the rejection. So it is due to cell mediated immune response, which is shortly termed CMI, that is responsible for graft rejection. All right. So this is the case. So that's why the correct answer is option A over here. Now, the next is question number 56. Now, question number 56 says that the bivalent or tetrad formation is a characteristic feature observed during. Now, this question has been asked from the chapter of your, cell cycle and cell division, all right, of standard 11. Again, this is also a direct question that has been asked. So bivalent or tetrad formation is one of the characteristic feature of zygotine actually, all right, that takes place. But if you just look into the options that has been given, so chiasma in the zygotine state, definitely this won't be the correct answer, because chiasma formation occurs in pectine stage. It basically occurs after the pectine stage, once the crossing over has taken place. That means in the diplotin stage. So you can say that chiasmata is formed in diplotin, not in the zygotine stage. All right. Now, the synaptonymal complex in the zygote. This is considered to be the correct answer because the bivalent is formed as a result of synapses. So, bivalent or tetrad formation is the result of synapses. So, this is usually the result of synapses and synaptonymal complex is formed as a result of synapses. As a result of this. All right. So that's why the correct answer is option B over here. Then chiasmata in the diplotin stage, again, no, we have to find out the bivalent. Bivalent means that there is a tetrad formation which is there. Then synaptonymal complex in the pectine stage, again, no, because synaptonymal complex is formed in the zygotine stage. So synapsis basically involves the pairing of homologous chromosomes. So synapsis involves pairing of homologous chromosome. That means one will come from the mother and the other will come from the father. So it looks like this. So a cell is having this one. All right. So this is bivalent, which is also called tetrad because there are 4 chromatids. All right. Now, the next is question number 57. So question number 57 says that you have to, the given below are the 2 statements, and one is labeled as assertion and the other is labeled as reason. So assertion A says that FSH which interacts with the membrane bound receptors does not enter the target cell. And the reason is that binding of FSH to its receptor generates secondary messengers, which is cyclic AMP, for its biochemical and physiological responses. All right. So you have to, in the light of the above statements, you have to choose the most appropriate answer from the options given below. Now, this question has been asked from the chapter of your chemical control and coordination of standard 11. All right. And let me tell you that the first option or the assertion which is given, A is basically considered to be the statement says that FSH, which it interacts with membrane bound receptor, does not enter the target cell. This is correct. So this is, the assertion is considered to be correct because we know that follicle stimulating hormone is actually a peptide hormone, which is a lipid insoluble hormone. All right. And lipid insoluble hormones cannot directly enter into the, they directly cannot enter into the membrane or this cell. Rather, they have to pass through the membranes by using the membrane bound receptors for carrying out their particular activity. All right. So we can say that so FSH is a basically peptide hormone. This is a peptide hormone, and the peptide hormone definitely cannot enter the cell because the membrane is made of lipid bilayer. That's why it's a peptide hormone, which is a lipid insoluble hormone that cannot directly enter the cell. So it actually cannot directly enter into the cell, and that's why it requires the membrane bound receptors. It requires membrane bound receptors. This usually requires membrane bound receptors for carrying out their activity. All right, so this statement is considered to be correct. While the reason that has been given is that binding of FSH to its receptor generates secondary messengers for its biochemical and physiological responses. Again, the reason is also considered to be true because definitely if the if the FSH will be binding it to its receptor, so the responses are produced in terms of, for example, like if this is a cell and this membrane, this is the membrane, all right, and this is basically the receptor which is present over here. So this is, consider this as the receptor. All right. So we know that FSH cannot pass the membrane directly. So this FSH molecule, which is present over here, all right, so this is going to first bind over here. Then this will initiate inside the cell. This is going to initiate some of the chemical reactions, and those chemical reactions are initiated as a result of the generation of the secondary messengers like cyclic AMP or CGMP in the form of all those things, or DAG, like that way. So induces secondary messengers. So definitely this is also considered to be as a correct reason. So R is correct because binding of FSH to receptor induces generation of secondary messengers. So this is going to induce the generation of secondary messengers. All right, for its biochemical and physiological responses. And physiological responses. All right, so that's why both of them, they are correct. But we can say that the reason is cannot be considered to be as the, you know, this one, that means the assertion which says that FSH which interacts with the membrane bound receptors does not enters the target cell. So this is considered to be as true. All right, and the reason is that binding of FSH to its receptors generates secondary messengers for its biochemical and physiological responses. So both of them are considered to be correct. Assertion is also correct, reason is also correct, but the reason is not the correct explanation of assertion, because the reason should be that if it is not entering inside the cell, so this indicates that it is a peptide hormone. The nature of the hormone is peptide hormone, and that's why it is not entering the target cell. We can say that option C is the correct answer. That means both of them are correct, but R is not the correct explanation of A. Correct. Option C is the correct answer. Now, question number 58. Question number 58 says that you have to choose the correct statement about a muscular tissue. So this question has been asked from the chapter of your animal tissues, and you just have to characterize the features of the muscular tissues that has been given, and you have to find out the correct statement. So the first statement, which says that smooth muscles are multinucleated and involuntary. So let me tell you that this is considered to be as incorrect statement. This is an incorrect statement because smooth muscles are uninucleate and involuntary. So smooth muscles are uninucleated, and along with that they are involuntary. All right, so definitely this is the incorrect statement. Then the next statement is skeletal muscle fibers are uninucleated and found in parallel bundles. Yes, this is a correct statement. All right, basically. But if you just look into the other statement, we'll also see the other statement as well. So skeletal muscle fibers, they are uninucleate and bundled together parallelly. All right. Now the third is intercalated discs allows the cardiac muscle cells to contract as a unit. All right. So this is again an incorrect statement because it is not the intercalated disc, but it is the communication junctions in the intercalated discs, which are called as gap junctions, that at some fusion points, is responsible for allowing the cells to contract as a unit. So we can say that gap junctions at intercalated discs of cardiac muscles allows cells to contract the unit. All right, so this is the case. Now, the option D is that the walls of the blood vessels are made of columnar epithelium. Now, let me tell you that the wall of the blood vessels, they are not made of columnar epithelium. Rather, they are made of, uh, epithelial tissue. All right, and that too, squamous epithelium tissue, you can say. The walls of blood vessels are made of simple squamous epithelium, not the column. All right. So that's why the correct answer is option B over here. Now, the next is question number 59. So the question number 59 says that you have to identify the region of human brain which has pneumotaxic center that alters respiratory rate by reducing the duration of inspiration. Right. Now, this question has been asked from the chapter of your breathing and exchange of gases of standard 11. And again, this is a very important question that has been asked. So let me tell you that pneumotaxic center is the center which is usually, which functions as respiratory rhythmic center. All right. So pneumotaxic center acts as respiratory rhythmic center. Now, since this is a respiratory rhythmic center, so this is usually found in the pons region of the brain. So, found in the pons region of, in fact, hindbrain. All right. So that's why out of all the options that you can see, the correct answer is pons over here. Now, the next is question number 60. So question number 60 says that the amount of biomass or organic matter which is produced per unit area over a given time period by plants during photosynthesis is called. All right. So this question has been asked from the chapter of your ecosystem of standard 12. And let me tell you that it is here asking about the total amount of biomass or the organic matter, and that too it is produced by plants during photosynthesis. We need to focus on this part. produced by plants during photosynthesis. So in productivity, you might have studied and heard about it, that there are two types of production in an ecosystem. One is called as the primary production, and the other is called as the secondary production. So primary production is considered to be as that production where, or primary productivity, in fact, you can say, is that productivity which is produced by plants only by the process of photosynthesis. So it is the total amount of biomass or organic matter produced by plants during photosynthesis. So technically the correct answer is option C over here, which you can see, primary production, because it is because plants only have the ability of fixing the sunlight or converting the light energy into chemical energy. So that's why it basically indicates that primary productivity indicates the total amount of biomass or organic matter which is produced by plants during photosynthesis over a given time period. All right. Now this primary productivity is further divided, into two categories. It can either be the gross primary productivity or it can either be the net primary productivity. All right. While the secondary production is the production which is produced by heterotrophs. So this is usually produced by heterotrophs. That means definitely not by plants which are autotrophs. And net productivity, net primary productivity is the productivity which is the available biomass for consumption to heterotrophs. So this net primary productivity is basically the available, or rather you can say, amount of organic matter available to heterotrophs. Heterotrophs. While on the other hand, the gross primary productivity is considered to be as the sum of net primary productivity plus respiration loss. Definitely, this cannot be the case. So that's why the correct answer is option D over here. So the next question is question number 61. So this question says that you have to select the incorrect match regarding the symbols that are used in the pedigree analysis. So this question has been asked from the chapter of your principles of inheritance and variation of standard 12. And let us take a look with the different options that has been given. So option A says that parent, the figure that has been given is parent with male child affected with disease. So definitely these are the parents. So this is considered to be correct. And this basically represents the affected child. And these represents the parent. So this basically represents parent. So this is considered to be as a correct match. All right. Now the next is sex unspecified. So this is also considered to be correct, which is represented by this one. So this is also correct. Then the next option is affected individual. So we have seen over here that if the box is filled with color or it is filled, so it is considered to be as an affected individual with any disease or something. So that is also correct. And the last is option D, which is consanguineous mating, which is represented by this. So again, this is considered to be incorrect because the consanguineous mating, which is known as the mating between relatives. All right. So this is being represented by, this option D is incorrect because this is being represented by this one. consanguineous mating. All right. So this is consanguineous mating, which is mating between relatives. Mating between relatives. Right. So that's why the correct answer is option D over here. D is considered to be as the correct answer because we have to find out the incorrect one. All right. Now, the next is question number 62. So question number 62 says that if the pH in the lysosomes is increased to alkaline, what will be the outcome? All right. So this question has been asked from the chapter of your cell, the unit of life, and it is asked from standard 11 of, and it is a question from the cell organelle lysosome. Now, let me tell you that lysosomes usually have an acidic pH. So naturally, lysosomes, they have an acidic pH because they contain many of the lysosomal enzymes like hydrolases and lyases, all those things, which works at that pH only. So we can say that normally lysosomes have acidic pH. All right. But if it is increased, but if the pH is increased to alkaline, but if pH is increased to alkaline conditions, then the major outcome, or you can say the major effect, will be that all the hydrolytic enzymes that are found in the lysosome that works in this acidic pH will be inactivated. So we can say that then the outcome will be all the, because these hydrolytic enzymes, as I told you, that includes, lipases or hydroxylases, that works at acidic pH will be inactivated. All right. So this is the case. That means out of all the options, if you will see, then lysosomal enzymes will be more active. Again, no. Hydrolytic enzymes will function more efficiently. No. Hydrolytic enzymes will become inactive. Yes, this is the correct one. And lysosomal enzymes will be released into the cytoplasm. All right. So that's where the correct answer is option C over here. The next is question number 63. So question number 63 says that according to the sliding filament theory, you have to find out the correct statement. All right. Now, a sliding filament theory, this question has been asked from the chapter of your locomotion and movement of standard 11. And in the process of sliding filament theory is basically related to muscular contraction. All right. Where there are different bands which are formed, A band, Z band, I band, all these bands are formed. So let us first write out that in sliding, let us try to understand the concept and theory first. So in sliding filament theory, the skeletal muscles, since this is related with muscular contraction, so that's why the skeletal muscles shortens during contraction. This usually shortens during contraction. because the actin filament is going to slide over the myosin, because the thin filaments, they are going to slide over the thick filaments. So because the thin filaments, and these thin filaments are being represented by A bands, which is active. Slide over the thick filaments, which is myosin. All right, that is the case. So thus we can say that the I band is going to reduce and the A band will retain its length. So this results into thus I band gets reduced and A band retains its length. All right. Now, as the thin filaments will be sliding inwards, the Z disc will be coming closer and the sarcomere is going to shorten. All right. Again, we can say that as the thin filaments, that means this actin filament I'm talking about, A band. So as the thin filaments slide inwards, the Z discs come close and sarcomere shortens. All right, so this is the, so let us take a look that what it is asking about. And let me tell you that this actin filament is basically the A band. Now, so the actin filament slides away from the A band, resulting in the shortening of the sarcomere. All right. The actin and myosin filaments slide over each other to increase the length of the sarcomere. Again, this is wrong. Then length of A band does not changes. Definitely the length of A band is not going to change. It is going to regain its own size. So this is technically correct. Now the first statement, it was left. So the actin filament slides away from the A band, resulting in the shortening of sarcomere. No. All right. Then the I band is going to increase in length. Again, the I band is not going to increase in length. Rather, it is going to get reduced. All right, and it will be getting reduced. So this is also the wrong statement that has been given. So that's why the correct one is option C over here. Now, the next is question number 64. Question number 64 says that the pathogenic bacteria gain resistance to antibiotics due to changes in them. All right. Now, this is actually a question that has been, it's a mixed kind of a question, you can say, that has been asked from the chapter of your biotechnology, preferably of your standard 12. And let me tell you that any bacteria which is pathogenic, that is going to gain the resistivity power towards the antibiotics because of their changes in the plasmid. So it's a very direct question that has been asked. The reason being is this, because plasmids are considered to be as the extra chromosomal circular DNA molecule, which is also known as the resistance plasmid actually. All right, so bacteria which shows resistance to antibiotic because they have the presence of our plasmids in them. That's why. So we can say that resistance plasmid which is also known as R plasmids allows specific bacteria to gain resistance against antibiotics. All right. So that's why the correct answer is option C over here. Now, the next is question number 65. So the question number 65 says that panspermia, an idea that is still a favorite for some astronomers, means. So this question has been asked from the chapter of your evolution of standard 12, because panspermia is also known as the theory of panspermia. And let me tell you, this spermia basically means spores. All right. And this, according to the theory of panspermia, the life originated on Earth because of the transfer of spores from one planet towards the another planet, and that's how the spores also got transferred to Earth as well, and that's how the life originated on Earth through these spores. So we can say that according to theory of panspermia, life originated on Earth due to spores, which was transported from one planet to another planet, just like one of the planet was Earth, which got transferred from one planet to another, in which, including Earth, rather, you can say, including Earth. Correct. It's because of the spores. So let us take a look. So transfer of spores as a unit of life from other planets to Earth. So this is considered to be the correct one. Now, creation of life from dead and decaying matter. This is called as the theory of abiogenesis. Actually, this is not the theory of panspermia. Rather, this is theory of abiogenesis, that which indicates that life originated from dead and decaying matter. Then the creation of life from chemicals. This is called as the chemical evolution theory, which is being indicated, or which indicates that life originated from chemicals. So this basically indicates organic evolution. This is related to organic evolution, or theory of chemical evolution, you can say. And origin of sperm in the human testes, again, this is also not the correct answer. So that's why the correct answer is option A. The next is question number 66. So question number 66 says that why CNG is considered better fuel than diesel. So this question has been asked from the chapter of your environmental issues of standard 12. And there are some statements given regarding it, that why CNG is considered to be better fuel. All right. So it cannot be adulterated. Definitely CNG, see, some of the advantageous features of CNG, which makes CNG, that means compressed natural gas, is a better fuel than diesel. So this is a better fuel than diesel and petrol, in fact, you can say, than diesel or petrol, due to certain reasons. And the first important reason is that it is cheaper. So it is cheaper as compared to diesel and fuel, and definitely it cannot be adulterated. So it can't be adulterated. and it burns very efficiently. It burns efficiently. All right. So this is the case. Efficient. So these are some of the reasons. So let us take a look. So it cannot be adulterated again. This is true. It takes less time to fill the fuel tank. No, this is not the reason. It burns more efficiently. Yes, it burns efficiently than petrol and diesel. That's why. So this is also true. It is cheaper. Yes, it is cheaper than petrol and diesel. It is less inflammable. No, it is highly inflammable. All right. So that's why this is also incorrect. So the correct answer is A, C, and D. So it's option C over here, which is the correct answer. All right. Now, the next is question number 67. So question number 67 says that which of the following statements are correct with respect to vital capacity? Now, this question has been asked from the chapter of your, breathing and exchange of gases, and it is regarding the different statements of vital capacity. All right. Now, let me tell you that what is vital capacity? So, vital capacity is considered to be as the, you know, this is basically the maximum volume of air that a person can breathe in after a possible forced expiration, or maximum volume of air a person can breathe out after a forced inspiration. So this is that way because it is a combination of ERV, TV, and IRV. You can say. So this is basically a combination of ERV, TV, and IRV. All right, which is expiratory reserve volume, tidal volume, and inspiratory reserve volume. So we can say that the maximum volume of air person can breathe in after a forced expiration, or maximum volume of air a person can breathe out after a forced inspiration. All right, so this is the statement. Now let us take a look. So it includes ERV, TV, and IRV. Yes, this is true. The total volume of air a person can inspire after a normal expiration. All right. So now, oh, okay. So you have to, this statement is also correct, but you have to choose, I guess, you have to choose the most appropriate answers from the options given below. Now you have to choose the most appropriate answer, remember. All right. Now, so A is also correct. Total volume of air a person can inspire after a normal expiration, not normal expiration, but forceful expiration. So definitely this is not the correct answer. Now, the maximum volume of air a person can breathe in after a forced expiration. Yes, this is true. Then it includes ERV, RV, and IRV. Again, no. All right. The maximum volume of air a person can breathe out after a forced inspiration. Again, this is also true. That means option A, C, and E is considered to be as correct. That means option D is the correct answer. All right. Now, the next is question number 68. So question number 68 says that how many secondary spermatocytes are required to form 400 million spermatozoa? All right. So for forming 400 million spermatozoa, so let us draw a figure first. So in case of the process of spermatogenesis, if you look into it, so it basically starts with a single cell, which is called spermatogonia. So spermatogonia, we all know, is a diploid cell. All right, so this is usually a diploid cell. And this ultimately divides and differentiates to give rise to primary cells like this, primary spermatocytes like this. So this is basically mitosis differentiation that you can see. It will undergo the process of mitosis differentiation to give rise to primary spermatocytes. These are all primary spermatocytes. Spermatocytes. All right. Again, this will also be diploid. Now, this primary spermatocyte will undergo the process of first meiotic division. That means it will undergo the process of first meiosis to give rise to two cells. All right. Like this. And that is called as secondary spermatocytes. So, since meiosis 1, we all know, is a reductional division. This is a reduction division. And since this is a reductional division stage, so definitely the ploidy of secondary spermatocytes will be haploid. This will be haploid. Now, this secondary spermatocyte will again undergo the process of second meiotic division. So, this will undergo the process of second meiotic division to give rise to 4 cells like this. So, this is meiosis second. And we know that meiosis second is an equational division. It will give rise to 4 cells. And these 4 cells are called as spermatids, which are again haploid. And these spermatids will again differentiate into, finally will differentiate into sperm. So each of these spermatids will be differentiating into sperm like this. So these are spermatozoa. Either you can say it as spermatozoa or sperm. So that means you can see over here that in the question it has been asked that how many secondary spermatocytes are required for 400 million of spermatozoa. So if from the 2 secondary spermatocytes, the 4 sperms are produced. So if from 2 secondary spermatocytes, the 4 sperms are produced over here. You can see over here. All right. So definitely if you have to produce 400 million spermatozoa, then your secondary spermatocytes have to be just half, which is 200 million. All right. So definitely option D is the correct answer, just half, because we all know that it will undergo the process of equational division like this, and so it will result into formation of spermatozoa. So option D is the correct answer. The next is question number 69. The question number 69 says that the mad cow disease in cattle and Creutzfeldt-Jakob disease in humans are due to infection by. So this question has been asked from the chapter of your biological classification of standard 11. And let me tell you that these two diseases which has been mentioned, these are very rare diseases which are caused by a very unique entity, and that is called prions. All right. So mad cow disease, so mad cow disease and Creutzfeldt Jakob disease. In humans is caused due to prions. Prions are considered to be as proteinaceous infectious particles which causes infections through their proteins because they lack genetic material. The proteinaceous infectious particles. They are called as proteinaceous infectious particles, which are actually a case of abnormally folded proteins, you can say. Abnormally folded proteins. All right, so this is the case. That means the option A is the correct answer. Now, the next is question number 70. So question number 70 says that you have to arrange the components of mammary gland from proximal to distal end. All right. Now, so let us arrange it from proximal to distal end. So the first component that will be coming up are the alveoli. All right. So the first will be alveoli. Alveoli will be followed by mammary tubules. So it will be mammary tubules. Then it is followed by your mammary duct. So it's mammary that will be carrying the passage. So it will be mammary duct. Mammary duct. Then after that, there is mammary ampulla or mammary ampullae is also called as mammary ampulla. And mammary ampulla will finally lead to lactiferous duct through which the milk is sucked out. Lactiferous. So this is the sequence. All right. So let us take a look into the options that has been given. So the options are alveoli. That means C. This is C. Then there is E. Then A. Then D. And then B. All right. So C, E, A, D, B. So C, E, A, D, B. This one. Option D is the correct answer. All right. The next question is question number 71. So this question says that Western Ghats have larger number of plants and animal species that are not found anywhere else. So which of the following term is used to notify such species? Now this question has been asked from the chapter of your biodiversity and its conservation of standard 12. And in that chapter, you might have heard it also that the Western Ghats usually have a greater number of plants and animal species as compared to any other ghats or areas. And the reason for this is that they have high number of endemic species. That means option D is the correct answer because endemic species are considered to be as those species which are confined only to a particular region and they are found nowhere else and they are not found anywhere else. So that's why the correct answer is endemic species, which is option 4. So endemic species are the species confined to a particular region or area and not found anywhere else. So Western Ghats are highly rich in endemic species of plants and animals, not found anywhere else. That's why the Western Ghats, they are rich in endemic species. Western Ghats are rich in endemic species. All right. So that is the case. While when you consider vulnerable species, threatened species, these are the species that belongs to red list category. So this cannot be included or red data book. So these are red list species. And on the other hand, keystone species is such species which is very less in number, but it might also be found that is controlling the community or the ecosystem functioning of that area. So again, this is also not an option. That's why the correct option is option D over here. Now, next is question number 72. So question number 72 is a match the column kind of a question that has been given. And with the listings that has been given in 1 and 2, you have to choose the correct answer from the options given below. All right. Now, this question has been asked from the chapter of from the animal type, which is cockroach. All right. The different structures of cockroach has been given, and you have to identify the functioning of it. All right. So the very first option is crop. Now, let me tell you that crop is found in case of cockroach, which is used in storage of food. All right, so this basically helps in, it's an organ that helps in storage of food. Rather, we can say an organ helps in storing food in cockroaches. All right, so this is going to match up with A. Then proventriculus. Proventriculus is a structure which helps in the grinding of the food particles. All right. So this basically helps in grinding the food particles. So this is going to match up with B. So whatever the food that will be stored up and will passed on to the, uh, you know, this proventriculus, which is also known as gizzard. So proventriculus or gizzard helps in grinding food. All right. Now, the third is hepatic CK. Now, hepatic CK is considered to be one such organ that helps in secreting the digestive juices. So, it will secrete the digestive juices. All right. So, hepatic CK. This basically helps in secretion of digestive juice. And that digestive juice will ultimately help in the digestion of the food particle. All right. And the last which is left is Malpighian tubules. Malpighian tubules is an excretory structure, or it is the excretory organ which is found in case of the cockroach that helps in removing the, you know, waste materials. So it helps in removal of nitrogenous waste. The D option is Malpighian tubule. An excretory organ that helps in removing excretory wastes. All right, now, so let us take a look into the options that has been given. So A is going to match up with fourth, B is going to match up with first, C is going to match up with second, and D is going to match up with third. That's what the correct option is, option B over here. Answer is B over here. All right, now the next is question number 73. So question number 73 says that two butterfly species are competing for the same nectar of a flower in a garden. All right. Now, to survive and coexist together, they may avoid competition in the same garden by. So definitely, this question has been asked from the chapter of your population and organisms, and population of standard 12. And let me tell you that this is one of the categories of population interaction. This question has been asked from the category of population interaction. So, in order to prevent or avoid competition, it is very important that these two butterflies, they should, choose their different foraging patterns. That means different feeding patterns will be there, so that competition coexistence will occur. All right. So coexistence will occur, and the competition will be avoided. All right. So we can say that to avoid competition, in the same garden, that is to prevent, you can say, competitive exclusion. That is to prevent competitive exclusion, two butterfly species competing for the same nectar of a flower should choose different foraging patterns. So if their foraging and feeding patterns will be different, so definitely they are going to coexist with one another. Foraging patterns that will promote their coexistence. Coexistence. All right. So this is the case. So predating on each other won't be the option because it will eliminate one species, and one species will be benefited and other will be harmed. Feeding at the same time, again, this will promote competitive exclusion. So definitely this cannot be the option. This is the correct answer. And increasing time spent on attacking each other, definitely no. All right. So that's why the correct answer is option C over here. Now, the next is question number 74. So question number 74 says that the role of enamel is to. Now, this question has been asked from the chapter of your human nutrition of standard 11, and that means digestion and absorption chapter. And the role of enamel is one of the hardest substance known in the body. All right. And this is usually found in the teeth portion, teeth part. The teeth covering is usually made of enamel. And it gives a hardest structure to the teeth, so that it will help in the mastication of food. All right. So that's why If you just look into the option which says that gives basic shape to the teeth, no. Connects crown of tooth with its root, again no. Masticates the food, yes. Forms bolus, again no. All right, because the major function is of enamel is to masticate only. So we can say that enamel is the hardest substance in the body found on the outer side of crown in teeth. All right, and this is usually involved, and because of this hardest substance, this is usually involved in mastication of food and helps in mastication of food. All right, the option C is the correct answer. Now, the next is question number 75. The question number 75 says that you have to choose the incorrect enzymatic reaction. Now again, this question has been asked from the chapter of your digestion and absorption of standard 11. Now different enzymes have been given acting on different substrates to give results. So let us take a look of, we have to identify the incorrect enzymatic reaction. So the first one, which says that the dipeptidases is responsible for converting the dipeptides into two amino acids molecules. So this is correct. This is usually considered to be correct because dipeptides, they get converted to amino acids this is usually correct with the help of dipeptidases. Now, the second one is maltose. In the presence of maltase, will get converted into glucose and galactose. Now, let me tell you that this is an incorrect statement because maltose is a disaccharide which, in the presence of maltase, will be converting into two molecules of glucose. All right, so this is considered to be as the incorrect enzymatic reaction. Let us take a look in the other as well. Sucrose, in the presence of sucrase, will get converted into glucose and fructose. Again, this is also correct. So sucrose is again a disaccharide molecule. In the presence of sucrase, will get converted into glucose and fructose, which are monosaccharides. And the last is lactase. Lactose actually. Lactose in the presence of lactase will get converted into glucose and galactose. All right, so that's why the incorrect statement which you can see over here is the option B. All right, now the next is question number 76. Now, in case of this question, which you can see, given below are the two statements. One is labeled as assertion and the other is labeled as reason. Assertion is that during pregnancy, the level of thyroxine is increased in the maternal blood. The reason given for this is that pregnancy is characterized by metabolic changes in the mother. Now, in the light of the above statements, you have to choose the most appropriate answer from the options given below. So, the assertion which says that during pregnancy the level of thyroxine is increased in the maternal blood. So the assertion is considered to be actually correct. A is correct because we know that during pregnancy, and this question has been asked from the chapter of your human reproduction of standard 12. So we know that in case of pregnancy, the metabolic rate in case of the pregnant mother, they that gets increased in order to fulfill the requirements of the mother as well as of the developing fetus, and that requirement is fulfilled by increasing the amount of thyroxine in the blood. All right, so that's why A is considered to be correct, that during pregnancy, the metabolic rate of mother is increased to fulfill the requirements, in order to fulfill the requirements of both mother and developing fetus. In order to fulfill the requirement of both mother and developing fetus, so that's why there is an increase in the level of the thyroxine as well. That increases the thyroxine in the blood. Thyroxine level in blood of mother. All right, in order to nourish it, basically. So this is a correct statement. While the reason that has been given for this is that pregnancy is characterized by metabolic changes in the mother. Yes, this statement we have actually seen it. It has been written here that yes, there is a metabolic changes in the mother that results into the changes in the level of the thyroxine in the blood as well. So definitely this is the correct reason. So we can say that R is also correct. That means reason is also correct. And in fact, the reason is considered to be as the correct explanation of assertion. So both A and R are correct, and R is the correct explanation of A. Option B is the correct answer. Now, the next is question number 77. Now, question number 77 says that you have to choose the correct statements. Bone supports and protect softer tissues and organs. Weight-bearing function is served by limb bones. Ligament is the site of production of blood. Adipose tissue is specialized to store fats. Then tendons attaches one bone to another, and you have to choose the most appropriate answer from the options given below. All right. Now let me tell you that option A, which says that bone supports and protects softer tissues and organs, this is a true statement actually. That yes, bones supports and protects softer tissues and organs. So this is true. This is considered to be as the correct statement actually. And the B option says that weight bearing function is served by limb bones. Yes, this is also true. That weight bearing function is served by limb bones. So this is also true. Now the next is that ligament is the site of the production of blood. No, this is a wrong statement. Ligament is not the site of the production of blood cells. It is rather the bone marrow, you know. So, ligament is not the site of blood production, rather it's bone marrow. So rather it's bone marrow. All right, so this is a false statement. All right, the D option says that adipose tissues is specialized to store fats. Yes, this is a true statement. Adipose tissues are fatty tissues which are responsible for storing fat. So adipose tissues stores fats. Now, the E option is that tendons attaches one bone to another. So tendons are not responsible for attaching bones to bones. Rather, it is responsible for attaching, you know, bones to ligament. So tendons attaches all right. So this is the statement which says that tendons attaches one bone to another. This is a false statement. This does not the tendons doesn't attaches one bone to another. One do not, we can say, tendons do not attach one bone to another. Rather, it's the ligament that attaches bones to bone and tendon attaches muscles to bones. Ligament that attaches one bone to another and tendon attaches muscles to bones. All right, so this is the case. Out of all the statements that you can see over here, so this is considered to be, that means option A, B, and D is considered to be the true statement. So A, B, and D. That means option B is the correct answer. All right. Question number 78. The question number 78 says that if DNA contains sulfur instead of phosphorus and proteins contain phosphorus instead of sulfur, if DNA contains sulfur instead of phosphorus and proteins contain phosphorus instead of sulfur, what would have been the outcome of Hersey and Chase experiment? Now, this question has been asked from the chapter of your molecular basis of inheritance of standard 12. All right. Now, this is one of the experimental setup that was put forward by Hersey and Chase. And let me tell you that in the question which says that if the DNA containing sulfur instead of phosphorus and the proteins is containing the phosphorus instead of sulfur, then what would be the outcome of the Hersey and Chase experiment? So if this would have occurred, then definitely the radioactive sulfur, it would have been found in the bacterial cells. All right. The radioactive sulfur would have been found in the bacterial cells, not the radioactive phosphorus, you can say. All right. So if DNA contain sulfur protein, sorry, if DNA contains sulfur and proteins contained phosphorus, then in this case, the radioactive sulfur which we have detected in the Hershey and Chase experiment would not that will be found in case of the bacterial cells. So the radioactive sulfur will be found in bacterial cells. So let us take a look into the options that has been given. So radioactive phosphorus, no. No radioactive sulfur, again, no. Both radioactive sulfur and phosphorus, no. Radioactive sulfur in the bacterial cell, yes. That means option D is the correct answer. Now the next is question number 79. Question number 79 says that you have to select the incorrect statement with respect to the cyclostomes. All right. So this question has been asked from the chapter of your animal kingdom of standard 11. And cyclostome is one of the categories of Pisces. All right. So let us take a look at the options that has been given. So the first option says that you have to choose the most appropriate answer from the options that are given. Now, cyclostomes, they lack scales and paired fins. This is true. All right. So cyclostomes, they lack scales and paired fins. So this is a correct statement. All right. Now they have circular mouth with, jaws. So this statement is considered to be as the correct. Now option B, which says that they have circular mouth with jaws. So yes, cyclostomes do have a circular sucking mouth or a suctorial mouth. So cyclostomes have circular mouth, but they lack jaw. They do not have any jaws, but lack jaws. That's why they are kept under Agnatha category. So this is definitely an incorrect statement. Now the next is option C, which says that they bear 6 to 15 pairs of gills. This is true. So cyclostomes bears 6 to 15 pairs of gills. They bear 6 to 15 pairs of gills. So this is true. And the D option is that says that they migrate to deep sea for spawning. No, this is a incorrect statement, because for the spawning purpose, or for the spawning, during the spawning period, for their spawning process, they usually migrate towards these cyclostomes. They are usually marine, so they migrate towards fresh water for their spawn. So they migrate to fresh water for spawning. So this is incorrect state. So with all the four options that has been given, you have to select the incorrect statement. All right. So the incorrect statement is option A, option B and D. So incorrect statements are B and D. Right. That means option D is the correct answer. The next is question number 80. Question number 80 says that a unique vascular connection between the digestive tract and the liver is called. So this question has been asked from the chapter of your digestion and absorption of standard 11, and it is asking about a unique vascular connection between the digestive tract and the liver. All right. So anything which is connected to liver is called as hepatic. And digestive tract is one such tract which is considered to be a specialized portal system. All right. So that's why it's a very direct question that has been asked, because hepatic cystic system is different. Hepatopancreatic system is between liver and pancreas. Renal portal system is with excretory system, that means kidney. All right. So definitely option C is the correct answer, which says that hepatic portal system is a specialized system that connects the digestive tract with liver. All right. So it's the hepatic portal system. Option D. All right. Option C is the correct answer. Now, the next is question number 81. Question number 81 says that the milk of transgenic cow, Rosie, was nutritionally more balanced product for human babies than the natural cow milk because it contained. So this question has been asked from the chapter of your biotechnology and its applications. All right. And we know that Rosie is one of the first transgenic cow which was being produced, and the milk that it produced was very, very rich in a very important protein, and that is called as human lactalbumin protein. All right, human alpha lactalbumin protein. So it's a very direct question that has been asked. So it's human enzyme ADA. No. So this is basically related. This ADA, that means adenosine deaminase, is related with immunity. So it has nothing to do with rosy or milk of transgenic cow. So that definitely this is the wrong one. Then human protein alpha 1 antitrypsin, again, this is also nothing to do with the transgenic cow. And human insulin like growth factor, again, no. So it is the only one that has been produced was highly enriched. The milk was highly enriched in the human alpha lactalbumin protein. So human alpha lactalbumin protein is the milk, or we can say this protein is found in the milk produced by transgenic cow Rosie. And it was nutritionally considered to be more balanced as compared to the natural cow milk. All right. So that's why the correct option is option C over here. Now, the next is question number 82. The question number 82 says that gout is a type of disorder which leads to. Now, this question has been asked from the chapter. This is basically asked from the chapter of your, locomotion and movement. All right. And of your standard 11. So let me tell you that gout is one of the metabolic disorders which is characterized or which happens because of increase in the level of the uric acid and uric acid crystals in the joints. All right. So this is a very characteristic disease, or it's a, you can say, a metabolic disease occurring due to deposition of uric acid and uric acid crystals. It occurs due to deposition of uric acid and uric acid crystals in the joints. In the joints, so leading to the inflammation in the leading to its inflammation. And so there's a difficulty in standing or walking or sitting, all those things. So weakening of bones due to low calcium level, no. So this is basically osteoporosis condition. So this cannot be the case. Inflammation of joints due to accumulation of uric acid crystals, yes. Weakening of bones due to decreased bone mass, again, no. Inflammation of joints due to cartilage degeneration, no. So option B is the correct answer. Now, question number 83. So which of the following methods is not commonly used for introducing foreign DNA into the plant cell? Remember. So this question has been asked from the chapter of your biotechnology, principles and processes. So there are various ways by which you can insert a foreign DNA either into a plant cell or into an animal cell. But the techniques are very different. In animal cell, the techniques are different. And in the plant cell, the techniques are entirely different. All right. So it is asking which is not commonly used for introducing DNA in the plant cell. You'll have to talk about it. All right. So out of all the options that has been given, all these three options, that means agrobacterium mediated transformation, then gene gun, which is also known as biolistics, all right, and then disarmed pathogen vector. So all these are used, for inserting foreign DNA into plant cell. But the bacteriophages, this is basically used for animal cell, not specialized for plant cell, but for the animal cells. Right. So you can say that bacteriophages, these are basically the viruses that infects bacteria. So bacteriophages are used to insert foreign DNA into bacteria. Why? Because this is a virus that infects bacteria. It's a virus infecting bacteria and not the plants. All right, so that's why the correct answer is option A. The next is question number 84. So question number 84 says that given below are the two statements. Statement 1 that says that amino acids have a property of ionizable nature of NH2 and COOH group. Hence, have different structures at different pH. Okay. And the statement 2 says that amino acids can exist at zwitterionic form at acidic and basic pH. So in the light of the above statement, choose the most appropriate answer from the options that are given below. All right. So that's, if you just look into this question has been asked from the chapter of your biomolecules of standard 11. All right. And if you just look into the statement 1, which says that ionizable forms of amino acids are available. So this is a correct statement actually. Statement 1 is considered to be correct because yes, ionizable forms of amino acids are nature, and we can like justify, the justification is like if I draw a simple structure of an amino acid, the ionizable forms are will be, or rather we can say this. Like this. All right. So this is basically the ionizable form. Now, this is basically the zwitterionic form, rather we can say. But if we have to draw the ionizable form, so we can draw like this if the pH is increased. All right. So if the pH, we can say, is less than 7. So if the pH is less than 7, so there will be the prevalence of the predominance of the hydrogen ion, and so the structure becomes like this. So it will be like this. Like this. So there will be an overall positive charge in the amino acid. So this is amino acid at pH 7, which is neutral. And just like that only, if we just consider the pH which is greater than 7. So in that case, the structure, there will be the prevalence of the OH minus ions. All right. And so the structure turns out to be as H2N, CH, there will be R, and then COO. So there will be the overall prevalence of negative ion. So these are ionizable forms which you can see. Ionizable. This is also ionizable. So this is having the presence of, you can see over here, negative, overall negative charge, and this is having the overall positive charge over here. All right. And this is the zwitterionic form. This is called zwitterion because it is overall having no negative net charges. So that's why it is called zwitterion. So yes, the statement is 1 is correct. And the statement 2, which says that amino acids can exist as zwitterionic form at an acidic and basic pH. Yes, at an acidic and basic pH. All right. So this zwitterionic form takes place at the isoelectric point that can be either at the acidic pH or at the basic pH. All right. So that's why both the statements, as you can see over here, both the statements are considered to be as the correct one. All right. So statement 2 is also correct. All right. That means both the statements are correct. Now, the next is question number 85. So question number 85 says that given below are the two statements. One is labeled as assertion and the other as reason. And you have to choose the most appropriate answer from the options given. Now, assertion says that spirulina is a microbe that can be used for reducing environmental pollution. The reason is that spirulina is a rich source of protein, carbs, fats, minerals, and vitamins. All right. So if you just look into the statements that has been given, so the assertion is true that spirulina, yes, spirulina is can be used for reducing the environmental pollution because it performs photosynthesis, it enriches the surrounding environment with oxygen. So that's why. All right. And it it grows in all the areas like it can grow in sewage, it can grow in waste waters, potato processing plants or industry straw, molasses, etc. So assertion is correct, which says that spirulina is a blue green alga that can be used for reducing pollution as it can grow on wastewaters, then potato processing plants. Basically, spirulina is one of the source of protein, one of the richest source of protein, single cell protein. Potato processing plants, so they can grow in all the waste areas, plants, then straw, molasses, etc. All right. So that's why this is considered to be as a correct one. All right. And spirulina is a rich source of protein, carbs, fats, minerals, and vitamins. So this is also considered to be as true that it is one of the richest source of proteins, which is considered to be. Basically, we can say that spirulina is an example of spirulina represents one of the alternate sources of protein. It basically represents one of the alternate sources of protein, that is single cell protein. It will be having more protein as compared to any other, things, as compared to other nutrients like carbs, fats, minerals, and vitamins. So we can say that reason is not correct. That means A is true, but R is considered to be false. All right. So A is correct, but R is not correct. Option D is the correct answer. All right. The next is question number 86. So this says that with respect to metaphase, which of the following statement is incorrect? All right. Now this question has been asked from the chapter of your cell cycle and cell division, and it is asking about the stage. All right, and particularly the metaphase. And if we just you people might have been knowing it as well, that metaphase is one such phase where all the chromosomes are aligned in an equatorial plane. So chromosomes aligned at equatorial plate. All right. So this usually gives an appearance like, you just see to it. So this is just an animal cell with the asterism drawing, spindle fiber attaching to the kinetochore part, and it is like this one. So equatorial plate is being there. All right. So this is the case. So let us first take a look into the options that has been given. So chromosomes lie at the equator of the cell. So this is actually correct. So this is a true statement. Then complete disintegration of the nuclear envelope takes place. Yes. Inside it, if you will see it, then yes, the complete disintegration of the nucleus envelope has taken place. This is also true and correct. Chromosomes are highly condensed. Yes. So this is also true. As you can see that these chromosomes, these represents chromosomes, and these chromosomes, they are highly condensed and very visible in case of the metaphase. And the last is that metaphase chromosomes, they are made of 4 sister chromatids held together by centromere. Now this is considered to be correct. Incorrect, because the metaphasic chromosome, as you can see, I'll just highlight this structure of chromosome. So chromosome is usually made of 2 sister chromatids and not 4 sister chromatids. So these two are 2 sister chromatids. And this is a metaphasic chromosome that I'm talking about. Two sister chromatids and not 4. All right, so that's why the incorrect statement is option D over here. So that's why the option D is the correct answer with regard to the incorrect statement. Now, the next is question number 87. Question number 87 says that against the codon 5 prime UAC, what would be the sequence of the anticodon on the tRNA? All right, so you will have to find out the sequence of anticodon on the tRNA actually. All right, so the mRNA codon, so let us just write it out, that the mRNA codon that has been given is 5 prime UAC 3 prime. This is the mRNA codon. So the tRNA anticodon, if you will talk about it, it is usually made of tRNA anticodon. This will be having a sequence of, if you just write it in sequence to this, then it will be 3 prime, 5 prime will be in against the 3 prime. You can like write it in a little closure way. Wait. So tRNA anticodon. The tRNA anticodon will be 3 prime A, U, G, 5 prime. This will be the case. So the sequence of anticodon on the tRNA, the sequence of anticodon on that particular tRNA will be considered to be as, if you just look into it, so it's this one. So it's 3 prime AUG, 3 prime AUG, 5 prime. This is considered to be as the correct answer. 5 prime AUG, no, 5 prime is coming towards the G, so definitely this is not the correct answer. T won't be there because it is the thymine part, so definitely the it is only found in case of DNA, not in case of RNA. So definitely C and D option will be the incorrect one. All right, so that's why the correct answer is option A over here. Now, the next is question number 88. Now, in case of the question number 88, you have to arrange the following formed elements in the decreasing order of their abundance in blood in the humans. All right. So, this question has been asked from the chapter of your body fluids and circulation and its circulation, standard 11. So, let us take a look into the different options. The first option is platelet. All right. See, we'll have to write it out in decreasing order. Decreasing order means that the highest one will be the first one. So if you just look into the options that has been given, so C will come first. That means erythrocytes, which are the RBCs, they will be having the maximum number. Erythrocytes or RBCs, they will be the maximum because they are known to have 5 to 5.5 million per millimeter cubic of blood. All right. Then comes the platelets, blood platelets. So these platelets, they usually have 1,50,000 to approximately 3,50,000, cells per millimeter cubic of blood. Then comes will be your leukocytes. That means WBCs. All right. So in case of the leukocytes, there are different categories that has been given. So neutrophils, lymphocyte, monocyte, eosinophil, and basophils. Different categories are there. So out of all the categories that you can see, the neutrophils will be the first one of them. So it will be B, which is neutrophil. Neutrophils, which usually have a percentage of around 60 to 65% in our body. Then comes after the neutrophils, it will be the eosinophils. Sorry, it will be the monocytes and then eosinophils. So monocytes is option E. So E is monocytes, which is having a percentage of 6 to 8. And then there is eosinophils, which is having a percentage of 2 to 3. All right. So that is the case. So if you just match the different options, so C, A, B, E, F. So it's C, A, B, E, D, C, A, B, E. Two options have been given actually. C, just a second. There is some, yeah, it shouldn't be F actually. This is not F. I have mistakenly written over here. This is actually D. The F option is not given. So it is D. So C, A, B, E, D. It's C, A, B, E, D. Option B is the correct answer. All right. Now, the next is question number 89. So question number 89 says that which of the following are true about the taxonomical aid key? Now this question has been asked from the chapter of your living world, very first chapter, living world of standard 11. All right. So let me tell you that the key is basically one such taxonomical aid which is based on similarities and dissimilarities having which are analytical in nature. So yes, we can say that keys is one of the taxonomical aid based on similarities and dissimilarities among features included. So whatever will be the features, so the similar and dissimilar features will be sorted. All right. So this is yes, this is basically analytical in nature. So they are considered to be analytical. So they are analytical in nature, and analytical in nature specifically means that it is based on two characters. That means keys are basically based on two contrasting characters that occurs in pair, which is called as couplet. All right. So this we can also say that they are based on contrasting characters. So they are usually based on contrasting characters called couplet, which usually occurs in pair. Occurring in pair. All right. So this is the case. Now, the next is same key can be used for all taxonomic categories. No, this cannot be the case. Definitely, this won't be the one because every character is different and it is based on contrasting characters. So, same key cannot be used. And each statement in the key is called as lead. Yes, this is also true. That each statement in a key is a lead. Each statement in a key is a lead. All right. So if we just look at the options, so option A is correct, then B is also correct, C, B is also correct, C is also correct, E is also correct, but D is false. All right. So that means A, B, C, and E are correct. A, B, C, and E. That means option D is the correct answer. Now, the next is question number 90. So question number 90 says that a normal girl whose mother is hemophilic carries a male with no ancestral history of hemophilia. So what will be the possibility of the phenotypes of the offspring? All right. Now this question has been asked from the chapter of your principles of inheritance and variation of standard 12. So let us draw a cross and try to understand that what would be the possibility. So according to the cross that has been given, the girl is normal. A normal girl whose mother is considered to be hemophilic. So mother is hemophilic, all right, and that mother is carrying that marries a male. That means hemophilic mother marries a male having no ancestral history of hemophilia. So definitely that male is considered to be normal. All right, male is considered to be normal. So what will be the possibility of the phenotype of the offsprings? See, if the mother is hemophilic, then definitely the girl will be a carrier. All right, that girl will carry that disease, so she will be a carrier. So we can say that normal girl whose mother is hemophilic will be having genotype which is this one. That means this she is receiving from a mother, received from a mother. All right, so that is the case. So let us draw a cross. So we can have this possibility and like this. All right. We could just look into the possibility. So this we can go with this one. And this we can go with this one, which is this. Okay. This will be XX and this will be X1. All right, so that is the case. So here will be the carrier daughter. Then this is affected male. That means hemophilic son, you can say. Hemophilic son. Because we know that hemophilia is a X-linked recessive disorder. Since hemophilia is a X-linked recessive disorder. That's why. This is a hemophilic son. Then again, this is a normal daughter. And this is a normal son. All right. So what is the probability if you see? So what will be the possible phenotypes of the offspring if we talk about it? So hemophilic son and hemophilic daughter will be there. Hemophilic daughter, as you can see, hemophilic son is there, but not the hemophilic daughter. So definitely this is not the correct answer. Then hemophilic son and carrier daughter, yes. Normal daughter, normal son, yes. Normal son and hemophilic daughter, no. So that means B and C are the correct option. That means option C is the correct answer. All right. Now, next is question number 19. So question number 91 says that in the enzyme which catalyzes the breakdown of H2O2 to H2O plus oxygen, the prosthetic group is. So basically H2O2. This question has been asked from the chapter of your biomolecules of standard 11. And this is called as hydrogen peroxide. This is usually called hydrogen peroxide. So hydrogen peroxide in the presence of enzyme peroxidase and catalase. So peroxidase and catalase, you can say, this gets converted into water and oxygen, useful products. All right. So in this category, the prosthetic group is basically known as the him. All right. So again, this is a very direct question that has been asked, which is option C over here. All right. So here, him is the prosthetic. All right. The next is question number 92. So the question number 92 says that you have to select the incorrect statement with respect to inbreeding of animals. All right. This question has been asked from the chapter of your strategies of enhancement in food production. So, with respect to inbreeding of animals, if it is asking about, and that it is asking about the incorrect statement. All right. So, it exposes the harmful recessive genes that are eliminated by selection. Yes, this is a true statement, and this is a correct statement actually. Let me tell you that inbreeding is one such process where there which involves the mating of closely related individuals, and that too within the same breed actually, merely for around 4 to 6 generations. All right. So inbreeding is the mating of closely related individuals within same breed for at least 4 to 6 generations. This is the prerequisite for, you know, inbreeding. And definitely, this inbreeding is responsible for exposing the harmful recessive genes. This exposes harmful recessive genes, then it basically results into creation of pure lines in the animals. So it creates pure lines in animals. So this is also true. This is also considered to be correct. All right. It helps in the accumulation of superior genes and elimination of the less desirable genes. So this is also correct. This is also considered to be true. All right. And the last is that it decreases homozygosity. No. If the purulines is increased, so definitely the chances of homozygosity will increase and not decrease. So inbreeding increases homozygosity. That can result into sterility also after some generations. So inbreeding increases homozygosity. All right. So that's why, with respect to the questions that has been asked, option D is the correct answer. All right. Now, the next is question number 93. Question number 93 says that IUDs are small objects made up of plastic or copper that are inserted in the uterine cavity. Now, this question has been asked from the chapter of your reproductive health of standard 12, let me tell you. And you have to find out the correct statement regarding IUDs. All right, that means intrauterine devices. So IUDs, which are also known as intrauterine devices, inserted by expert doctors or nurses in the uterus of females as a contraceptive method. All right, or as a contraceptive device, you can say. Now, these IUDs, they are of three types. Okay. They are of usually three types. So these are of usually three types. They can be hormone releasing IUDs, copper releasing IUDs, and non-medicated IUDs. So copper releasing, then non-medicated, and the last is hormone. So each one of them is having its specific function actually, and specific target hormone. So copper releasing basically includes copper T, then copper 7, then multi-load 375. Because of which multi-load 375. So this is basically, copper ions are basically responsible for suppressing sperm motility. So copper ions suppresses sperm motility. All right, so this is the case. Non-medicated IUDs basically includes this category of non-medicated IUDs. Non-medicated IUDs basically includes lipase loop. And hormone releasing IUDs includes Progesta Cert and LNG20. All right, so let us first take a look into the options that has been given. So IUDs decreases phagocytosis of sperm within the uterus. It doesn't decreases, rather it increases. All right, so this is the wrong statement. IUDs are basically responsible for increasing phagocytosis. So it increases phagocytosis of sperms. All right, so this is the case. Then the released copper ion suppresses sperm motility. This is true. IUDs do not make the cervix hostile to sperm. This statement is considered to be, let us take a look at the other statements also. So IUDs suppresses the fertilization capacity of the sperm. Yes, since the sperm motility is reduced, so definitely the fertilization capacity is also reduced. And the last is the IUDs require surgical intervention for their insertion into the uterine cavity. No, it doesn't require any kind of surgical instrument or something. So definitely this is also wrong. So the only one statement is IUDs do not make cervix hostile to the sperms. It actually makes cervix. Technically, it is, since the IUDs will be passing, will be inserted into the uterus. So definitely, whatever the sperms that will be entering inside the female reproductive structure or system, that will first have to go through the cervix part. It makes the cervix hostile to the sperm. So that's why it's the incorrect one. So out of all the options that you can see, the most appropriate answer is option D over here, which is B and D. This one. All right. Now, the next is question number 94. Question number 94 says that you have to select the correct statement regarding the mutation theory of evolution. All right. Now, this question has been asked from the chapter of evolution of standard 12. Now, let me tell you that mutation theory was the theory which was given by Hugo de Vries, and he basically contradicted the Darwinian concept of natural selection. All right. And according to the mutation theory of evolution, De Vries said that mutation is responsible for causing, changes in the population, which is random and directionless. All right. So we can say that mutation theory given by Hugo De Vries explained. It basically explained that mutation is the large differences arising suddenly in population. This is arising suddenly in population. That means single step, large mutation, you can say. All right. That is single step, large mutation concept. And that the mutations are random and directional. All right. So let us take a look at the option. Large differences due to mutations arise gradually in a population. Doesn't arises gradually, but it arises suddenly. So definitely this is wrong. This theory was proposed by Alfred Wallace. No. Variations are small directional changes. Again, no. Then single step large mutation is a cause of speciation. Yes. This is the correct statement, which says that single step large mutation, which will promote for speciation. All right. So option D is the correct answer. Question number 95. Now, 95 says that excretion in cockroach is performed by all except. Now, this question has been asked from the chapter of your animal type, that means animal tissues and animal type cockroach. So, let me tell you that excretion is performed by all these three. All these three are responsible for performing excretion. That means uricose glands, Malpighian tubules, and fat body. All right. All these performs excretion in cockroach. However, hepatic cecay is basically responsible for releasing digestive juices in the junction of foregut and midgut so that it will help in the process of digestion of food particles. So, hepatic cecay, which is also called as gastric cecay. Hepatic cecay releases digestive juices at the junction of foregut and midgut. All right, so this basically releases digestive juices at the junction of foregut and midgut, and this is basically a ring of 6 to 8 blind tubules. All right, so this has nothing to do with excretion, rather it is related with digestion. That's why option A is the correct answer. Now, the next is question number 96. Question number 96 says that you have to select the correct statement. Angiotensin second activates the cortex of adrenal gland to release aldosterone. Then The second statement is the next is aldosterone leads to increase in the blood pressure. ANF acts as check on renin-angiotensin mechanism. ADH causes vasodilation. Vasopressin is released from the adenohypophysis. All right. Now, sorry. Now, this question has been asked from the chapter of your chemical control and coordination of standard 11 regarding the different hormones. Now, let me tell you that this, out of all the statements that has been given, all right, so it is asking about ADH. So, hypothalamus is responsible for releasing the antidiuretic hormone, all right, which we can say it as hypothalamus releases ADH. Which is also known as vasopressin. All right. And that too to the neurohypophysis part. There are two parts, adenohypophysis and neurohypophysis. So through the posterior pituitary part. The neurohypophysis. All right. And this antidiuretic hormone is one such hormone that acts on kidney, responsible for resorption of water and electrolytes from the kidney tubules. This, rather we can say that this, the options if you see, so that has been given is angiotensin 2 activates the cortex of the adrenal. You have to choose the most appropriate answer. All right, remember. So the angiotensin 2 activates the cortex of adrenal gland to release aldosterone, and aldosterone leads to increase in the blood pressure. This is true. This is considered to be as a true statement. All right, this is true. Then ANF, that means atrial natriuretic factor, acts as a check on renin-angiotensin mechanism. This is again also true. All right. Then ADH causes vasodilation. Antidiuretic hormone is responsible for dilating the blood vessels. So this is also considered to be as true. Then vasopressin is released from the adenohypophysis. This statement is wrong. This is false. So we have found out our statement. That means A, B, and C is found to be as correct, which is option A. All right. So we can say the statement as ADH acts on kidney and affects its functions by its constrictory effect on blood vessels. All right. So this is the case. Option A is the correct answer. Now, the next is question number 97. Question number 97 says that if A and C makes 30% and 20% of DNA, respectively, what will be the percentage composition of T and G? Now, this question has been asked from the chapter of your principles of inheritance and variation, and it is based on Chargaff's rule. So, according to Chargaff's rule, if we say, then we all know that A is going to make double hydrogen bonds with T, and G is going to make triple hydrogen bond with C. All right, so that is the case. So if A is having a percentage of 30 and C is having a percentage of 20 in DNA, then definitely both will be equivalent. So A will be equivalent to T. So that means T will also be 30% and C will be bonding with G. That will be again equivalent to 20% in DNA. So we can say that G is 20% and T is 30%. So T is 30%, G is 20%. That means option C is the correct answer. All right. Now, the next is question number 98. The question number 98 says that refer the following statements for agarose gel electrophoresis. Now, this question has been asked from the chapter of your principles of biotechnology, principles and processes of standard 12. All right. Now, let me tell you that option A, which says that agarose is a natural polymer which is obtained from seaweed. Yes, this is true that agarose is a natural polymer which is obtained from seaweed. So this is considered to be as a true statement. All right. Now, the separation of DNA molecules in agarose gel electrophoresis depends on the size of the DNA. Yes, this is also considered to be true. Basically, let me tell you that agarose gel electrophoresis is one such method which depends on the separation of the DNA fragments on the basis of phoresis, which is usually based on the separation of DNA fragments on the basis of their size on passing current to it. So, separation of DNA fragments on a gel matrix on the basis of size by passing electric current. All right, so this is the case. So the DNA migrates from negatively charged electrode towards the positively charged electrode. Yes, we all know that DNA is negatively charged, so it will be definitely migrating towards the positive charged electrode. All right, which is the anode. So again, this is also true because it is because of the passage of due to passage of electric current it is talking about. The DNA migrates from positively charged electrode towards the negatively charged electrode. Now this is wrong. All right. No, we know that DNA is negatively charged, so it will migrate towards positive charge because opposite poles are going to attract. That's why. So the correct answer is A. The options are A, B, and C. So it's option C is the correct answer. Now the next is question number 99. The question number 99 is a match the column question that has been asked, and it is from the chapter of your locomotion and movement only, and specific part from the 11th section. And let us talk about it. So the first option given is multipolar. Multipolar. So multipolar basically means multipolar neurons it is talking about. So multipolar neurons means that it will be having one axon with two or more dendrites found in the cerebral cortex. All right. So multipolar means one axon with two or more dendrites. And this is found in cerebral cortex. All right. Now, the B is bipolar neuron. Bipolar neuron means that it will be having one axon and one dendrite. So, bipolar means one axon and one dendrite. And this is found in the retina of the eye. The example is retina, which is this one. Then there is myelinated nerve fiber. Myelinated nerve fiber is found in the spinal and cranial nerves, which will promote faster conduction, impulse conduction. So it is usually found in spinal and your cranial nerves, you can say. That means this one. Myelinated nerve fiber is found in spinal and cranial. The last is unmyelinated nerve fibers, which are found in the somatic neural system. All right, autonomous and somatic neural system. Smile. All right. So let us match with the options. So A is going to match up with second. B is going to match up with third. C is going to match up with fourth. And D is going to match up with first. That means option A is the correct answer. The last question of this paper is 100th question, which is given in the form of match the column. Right. So let us take a look into the options that has been given. So this question or the options that you can see is basically is from the chapter of human health and disease of standard 12. All right. And the first option says cellular barrier. All right. Cellular barriers. So the examples of cellular barriers are basically the PMNL, which is your polymorphonuclear leukocytes. So this is PMNL, which is polymorphonuclear leukocytes. So this is PMNL. All right. So this is one of the category of you can say neutrophils. All right. So this is a category of neutrophils. This is going to match with A. Then there is cytokine barrier. Cytokine barrier is an example of interferons. All right. One of the best examples. So cytokine barrier. Example of cytokine barrier is interferons. And interferons are basically a kind of a proteins which are being released by the virus infected cells. So proteins released by virus infected cells. which will actually prevent the other non-infected cells from getting infected. Virus infected cells, which prevents, or rather we can say which protects non-infected cells from getting infected. All right, so this is the case. That means the then the option C is physical barrier. So physical barrier is mucus. All right. The example of one of the physical barriers are mucus, which is released in epithelial linings, then respiratory tracts, all those cases. So physical barrier is, apart from mucus, it also involves tears and all those things. So physical barriers include skin also and mucus. Skin and mucus. These are the main categories. All right. And the last is physiological barrier. So in case of the physiological barrier, the last option which is left is the HCl, which is released in the gastric juice that prevents any kind of, bacterial infection in the stomach. So HCl in gastric juice. All right, so let's match up with the options that has been given. So A is going to match up with third, B is going to match up with first, C is going to match up with second, and D is going to match up with fourth. That means option A is the correct answer. All right. So this was all about the question paper of NEET 2022 that we have successfully completed and solved it. All right, and so that's all. So we will be doing the same with other NEET papers as well, other competitive exam papers as well. So till then, keep learning and keep scoring high. And for any kind of doubts that are related to papers, please join our Telegram channel link that is given in the description section. All right. So keep learning and keep scoring. Thank you very much.